So welcome to Lunch with the Core. Feel free to keep getting pizza here for the next couple minutes. Today's speaker is Moonsub and from the Don Ork Lab, and he's going to tell us about exploring chloroplasts of C4 plants with microscopy. He's been using the LSM 880 and also the serial block-based SEM with keys. So welcome.
Yeah. Could you turn off the light? Yeah, okay, thank you. Thank you, and hello, thank you for inviting me here at this research seminar, and I'm very appreciative. I thought that just only a couple of people are coming today, but too much people are coming, so I making a little bit feel nervous. So, yeah, my name is Moonsub Lee. I'm a postdoc working in Professor Donald Ork Research Group at the University of Illinois. And today I'm going to talking about one of my research, and specifically I'm going to talk about the C4 plants chloroplasts with using the SBF SEM and LSM microscope. So hopefully after this seminar, it might be helpful for your research or selecting the microscope. So I'm working in Don's lab, and then I'm working with the C4 grass species. Specifically, I'm studying on the photosynthesis to improve the crop yield, crop productivity. And so this is the figure showing how I measure photosynthetic, and then how I'm going to studying with the how I measure the photosynthetic efficiency in the greenhouse system. So photosynthesis is critical for the plant, and also photosynthesis has a significant impact on the crop productivity. So I believe at least no one doubt about the importance of photosynthesis. So, and regarding the photosynthetic efficiency, and fluctuating light or changing light intensity in the light level at the changing in light intensity at the leaf level is known as a factor to reduce the photosynthetic efficiency. So when you think about plant grow in the field, plant inevitably exposed to the fluctuating light caused by the passing cloud or changing in sun angle or the crossing the leaf. So let's look at this figure. First, the panel A. The panel A is showing the change in light intensity during a day. So in the panel A, x-axis indicates the time, and y-axis indicates the light intensity change from 0 to 2000 p50. So as you can see in the panel A, light changing during a day, this light intensity from 0 to 2000 is continuously changing. And also when you look at the panel B is showing the how much photosynthetic rate changes during the fluctuating light. So in the panel B, x-axis indicate time and y-axis indicate photosynthetic rate, and this solid line indicates the expected photosynthetic rate. This dotted line indicates the actual observed photosynthetic rate. So as you can see in the panel B, during the fluctuating light, you can see there is reduction in photosynthetic efficiency. This is because light change can be quickly, but photosynthetic rate adjusts slowly, which ultimately affects the crop productivity. So to improve the photosynthetic efficiency on the fluctuating light, our research group trying to modify the chloroplast, specifically the chloroplast volume. So you may wonder why we want to modify the chloroplast volume, but so this is all based on previous research. So actually, I really love to share whole of this background information and whole of our hypothesis, but due to the time limitation and today's the seminar's purpose, I'm not going to talking about it. So if you want interesting about the story, you can please let me know after this talk, or you can see the further information in our recently publication in this paper. So going back to the slide, as I mentioned earlier, our research group wanted to try to modify the chloroplast volume, specifically the chloroplast size. So we targeted diverse of gene involved in chloroplast division. So as you can see, this is the example of the genes that involve the chloroplast division. So we modified the gene, and we just changed the gene regulation, and our research group successfully generated transgenic lines. And then I was so happy, but I got the shock because our research group generated more than 200 the transgenic line. And then here is the problem: how can we measure those chloroplasts of the CPO plant? That's the big challenging for me at that time. So I tried to diverse the microscope, but when you think about the traditional microscope analysis, it's all based on two-dimensional analysis. So when you think about chloroplast cell, they are all three-dimensional objects. So based on two-dimensional analysis, it can result in the biased information. Also, this is actually, I took a picture using in the Beckman Center in TEM, transmission electron microscope, and just a regular optical microscope. As you can see, it's very hard to get the nice image, and also this is worse in the optical microscope. So it's making me getting hard to further analysis. And also adding more, here's another problem, because I'm working with the CPO plant. So you might wonder why CPOs matter. To explaining these things, I want to show briefly, very simply explaining about the CPO and C3 photosynthesis CPO C3 plant. So when you look at the C3 plant, in C3 plant, including soybean, wheat, rice, they capture CO2 in the mesophyll cell, and then they do the photosynthesis in the mesophyll cell. I'm very generally simplified the story. But in case of the CPO plant, it's totally different story. In CPO plant, including sugarcane, maize, and sorghum, they have two different cells. One is mesophyll, and the other one is bundle sheath cell. So for example, in CPO plant, they capture CO2 in the mesophyll cell with they capture CO2, and then they raising the CO2 concentration by using their specific CPO enzyme, and then send it to the bundle sheath cell, and then they do the photosynthesis, and they make glucose. So here in this slide, I just want you guys to remember only one thing, not that this thing, just only one thing I just let you know. Remember just only one thing. There is CPO plant has two different cells. One is a mesophyll cell, the other one is bundle sheath cell. In other words, there is two different chloroplasts in here, mesophyll chloroplast and bundle sheath chloroplast. So this thing is another make me hard to analyze the chloroplast, because how can I distinguish between the two different cells, and how can I exactly measure this issue? So I spent 1 year trying to find a better idea or better estimation method. And I'm very frustrating. I'm very almost exhausted. But one day, he is the Kingsley. He is the senior researcher in our core facility. He saved me. So going back to the slide. So one day he suggests me what about using the serial block phase scanning electron microscope? Hereafter, I'm going to call SVF-SEM. So in this experiment, so we wanted to try to establish a baseline information on the CPO plant chloroplast, because despite of the importance of the chloroplast, CPO chloroplast, little is known about this information. And moreover, in this experiment, we want to develop the kind of the fast screening method, the estimating chloroplast method. So we use two cutting-edge microscopes. One is SVFM, and the other one is the LSM 8800 with the area scan. Both of them are provided by the IGB core facility. So first of all, I will show the SVFM. I'm going to briefly introduce the SVFM, the inside chamber. So SVFM, inside of this microscope, there is a fine diamond blade. So what is this working, or what the SVFM works? Firstly, inside the SVF-SEM, repeatedly and cut the sample, and then capture images by this SEM. So SCBF SEM can provide more than hundreds of high resolution two-dimensional images that ultimately used for constructing the 3D image. So I will show one of our image. So this is one of the image I got from the SCBF SEM. So in this experiment, we just capture 500 slides at the 0.05 micrometer depth. So as you can see, I was very satisfied this result because I can see the clearly separate two different chloroplasts. And even when you look at the bundle shear cell, if you magnify this image, you can see the starch and you can see the grana, and all over you can see the anatomical feature. So then how are we gonna make a 3D block image? Let's look at the short videos. This is showing how I can make from individual 500 slides, just make one big 3D image. So that's the image block. So I'm trying to capture, condense, make one 3D block image. So this is image from obtained from SCBF SEM from the entire raw vlog images, and then from the entire raw data, and then this is process one. So let's look at detail individually. So the panel A is showing just like entire 3D block images, raw data. So this raw data image is too big, and there are so many huge and big data, so we cannot process it. So basically, based on your interests, we just set a separate extract sub-block, sub-volume. So in case of the panel B, showing the bundle shear cell sub-block, and then panel C is showing the mesophyll cell bundle shear, mesophyll cell chloroplast sub-block. So based on those sub-block, I'm processing with the 3D segmentation work, and then I can get individual clear image from the 3D visualized chloroplast image. So panel D is showing the bundle shear chloroplast, and panel E indicates individual mesophyll chloroplast. Let's look at the more detailed information about the chloroplast volume. First, panel B is showing the individual bundle shears and mesophyll chloroplast volume. As you can see the chloroplast, the shape is not the sphere. It looks like more ellipsoid shape, exactly more narrow type of ellipsoid, the shape. And then as you can see, like visually, you can see something, you can feel there's some different size between the mesophyll and bundle shear cell. So let's move on to the panel B. Panel B is showing the relationship between the chloroplast volume and the surface area. And red color indicate the wheat mesophyll cell, and green rectangular color is to indicate the chickpea. And then chickpea and wheat is also both two species, the C3 plant. And yellow color indicate the sugarcane, which is C4. So yellow circle, this one is the sugarcane mesophyll cell, and this yellow color indicates the sugarcane bundle shear cell. This graph, actually, there are so many information, but in this slide, I just want to you guys to remember only two things. First thing, just comparing the volume, mesophyll volume size, sugarcane mesophyll versus the bundle shear cell chloroplast. So bundle shear cell chloroplast, and sugarcane, and much bigger, 2 or 3 times bigger than mesophyll cell as we expected. And also sugarcane mesophyll cell, when compared to the other C3-2 plant, our sugarcane mesophyll cell chloroplast volume is smaller than the C3 wheat and chickpea, the mesophyll cell. Why these things happen, or is there any reason? So possible one of the explanation is, in case of a C4 plant, it might be beneficial for C4 plant to having a small or fewer the mesophyll cell chloroplast to avoid shading the chloroplast in bundle shear cell, which is also required to the light. So if we're assuming if SIPO plant have a large and packing inside if they have a packing in large in mesophyll chloroplast, that significantly impacts on the shading on bundle cell. So that's the reason why the SIPO mesophyll is smaller than the C31, and also why they have a more bigger and bigger chloroplast in the bundle cell. It's a possible explanation. And then I thought that's enough information for my paper, so I decide I'm presenting in our lab meeting, and then all our lab member responses. What about cell and other information? So I need to dig up. I spent 1 month in the imaging room from 9 to 12. And then finally, actually, last Friday, I finished all of the cell information. So that's when you think about the plant cell, that's the common image. I just Googled it. And also I thought, when thinking about plant cell, just looks like that, very cute, very nice sphere cells, and you can see chloroplasts and peroxisome and mitochondria. That's what you exactly expect. But in reality, it looks totally different. This is one of the examples I visualized the bundle sheath cell chloroplast. But panel A is showing the bundle sheath chloroplast. They look like the cylinder, looks like Pringles can. So they look 3D. They look like cylinder can. Also, across, I just testing the sugarcane and maize. They all look like same. They look like cylinder, no variation. But when you look at the panel B, the mesophyll cell, the shape is totally different. So when I showing this mesophyll cell to my advisor, he said it looks like the sculpture in front of our IGB, the building. Let's see it's true or not. So I'm prepared some more videos, so let's look at it's true or not. So that's the 3D image for when you look at the mesophyll cell. They look like bumpy and very low. It's very hard to explain their shape, but I will talk a little bit later why they look like this, like a very sharpy and bumpy and very hard to explain. So also I bring another slide, because whenever I show this mesophyll cell, all of our lab members say, is it true, Moonsub? So I take a multiple cell image in the sugarcane and maize. So A is showing the mesophyll cell in the sugarcane mesophyll cell structure, and B is showing the maize mesophyll cell structure. As you can see, they look like very weird and seems like very sharpy and bumpy and low. I don't know, it's very hard to explain this shape. I will talk to later, but this is the reason is because the mesophyll cell was very tightly covered the chloroplast. That's why they look like weird pattern. I will show the inside of what they look like. So one of the benefit of this IGB co-facility, I can access about the very fancy 3D program. So I have information about the individual chloroplast information, and also I have a cell wall information. So I can combining two information in one image, and then I can look at just like cut it half and then look at how they look like. That's one of the example what bundle sheath cell look like, what bundle sheath with the chloroplast look like. This is one of the example the bundle sheath cell. So let's look at. So this is green color is bundle sheath cell, and this is the gray color indicate the bundle sheath cell wall. So this is more detailed information. Panel A showing the bundle sheath cell with the chloroplast in front of the front view, and the panel B showing the top of view in the bundle sheath cell with the chloroplast. So let's look at first the bundle sheath cell bundle sheath cell with the chloroplast in panel A. As you can see, the panel A, bundle sheath cell chloroplast, they are very highly packing, and they packed in the bundle sheath cell, and they are very compacting, very tightly putting in each other, and approximately within our sampling depth in per 25 is bundle sheath cell counting in the one bundle sheath cell. And also when you look at the panel B in the top view, when you look at the panel B in the bundle sheath cell and chloroplast, there's something like looks weird. All of the chloroplast distribute to the left side. Why they look like? What's the reason? So one of the possible explanation is this is the leaf cross-section image in the maize. Red color is indicate the bundle shear cell, and blue color indicate the mesophyll cell. So as I mentioned in the first slide, I just talked to you guys to remember only one thing. Sepal plant has a two cell. One is the mesophyll and bundle shear cell. So they're trying to exchange metabolite. So that's the reason bundle shear cell chloroplasts, they are distributed near to the mesophyll cell. Thereby they can more facilitating the exchange of their metabolite. That's one of the reasons why the bundle shear cell, they are distributed into the mesophyll cell area. And what about the mesophyll cell with the chloroplast looks like? I like to see this video. I can see it all day, so let's look at for this video quick. This is the gray color is also mesophyll cell wall, and green color is chloroplast. This is part of the mesophyll cell. So yeah, this is the mesophyll cell chloroplast, and this gray color is the cell wall. First time when I look at the image, I was surprised because the cell wall is very highly covered the mesophyll cell. That's why we saw the very weird shape, because they very highly, exactly, they looks like they are wrapping the chloroplast. That's why look at some different shape. And also in case of a C3 plant, it's not C4, just in C3 plant, if they're closely packing the cell wall and chloroplast, there is some benefit in C3 plant because the CO2 came out from the intercellular space and they go penetrate CO2 into the chloroplast. If the distance is shorter, it's more beneficial. But in case of CPO, we were surprised because CPO plant, I just mentioned earlier, they have a unique enzyme. They don't need to, they have some enzyme to keep concentrating and increase CO2 level. So we are just still wondering why this chloroplast, the cell were very close packed. And also, when I compare to the cell wall, this other previous publication, this is the cell wall 3D visualized the mesophyll cell wall in sugarcane and this maize one. And this is also not the same microscope. This is published in 1995. They're using the micro XT, the microscope. They just visualizing their C3 plant, the mesophyll, the cell. They looks like different, and ours is more the Sharpie. This is, we assuming this is related to the surface area. So when you think about the volume versus surface area, even though the same volume, but depends on the shape, the surfaces can be changed. So this might be some benefit for the surface area into the intercellular space, but still we are discussing about these things, what they look like, what they look like. So if you have any idea, just let me know after this talk. So because we still we don't know about it, so we're still discussing items. So in this current situation, so another benefit in the SBF-SM, they provide very high resolution picture. So I just keep showing the leaf cross section. But I just mentioned in earlier, bundle shear cell and mesophyll cell, they connect and they cooperate in each cell. So when I close this area, we can see the plasma desmata. So you might be some people are not familiar with the plasma desmata. So plasma desmata is a channel, the connecting channel between the bundle shears and mesophyll cell. And SBFM is, I was surprised because they very clearly capture this very tiny, the plasma desmata channel. And also, I can see the only not XY view, and also, but also I can see the Y different view, and the more clearly how they're connecting, we can see this area. So, in our research team, Ryan and I, we decide to visualize this plasma desmata between this bundle shear cell. So this is one of the example. This is the bundle shear cell, chloroplast and bundle shear cell, and those entire, the feed hole is showing the plasma desmata as bundle shear cell. And Ryan, he's our lab postdoc. He's more ambitious than, so he's make a more clear image about the plasma desmata. Let's look at this figure. this is showing the cell wall. The yellow color indicates the plasma. Yellow color indicates the bundle sheath cell wall, and blue color indicates the one over the next to the mesosphere cell. The green color is also mesosphere cell, connecting each other cells. And then Ryan trying to cut this subsample and trying to visualize it. So this individual hole indicates plasma desmata kind of area, and also this different light blue color showing some different color in this mesosphere cell. So why do you want to do this job? Our main goal is trying to kind of quantify how many plasma desmata hole in each cell, and how many cells, how many plasma desmata feed hole is located, and how much they are closely connected. So we want to kind of counting the cell. So let's look at this figure, how they look like. So this is the yellow is the bundle sheath cell. This is connecting the one mesosphere cell, the green color. This one has some pink, is indicate the plasma desmata. And this blue color is also indicate another mesosphere cell. They are two connecting each other. And blue is also another mesosphere cell connecting. So you can see that there are a lot of holes. So this is a partial image. So we have data. So I just trying to put all of information like a Power Ranger robot they assemble. So I just put the bundle sheath cell with the small plasma desmata feed hole, and then put him in the mesosphere cell. Mesosphere also have a bundle sheath cell hole. Just like put him all together and just like a visualize 3D image. And then let's look at how they look like. This is one of the example in maze. So before starting, this 3D cylinder is the bundle sheath cell, and this one is plasma desmata in the bundle sheath, and this one is the gray color, is the mesosphere cell. So yeah, this, as you can see, because some bundle sheath cell has some plasma desmata, this is connecting to another bundle sheath cell. That's why we can see it. This is attached to another one of the mesosphere cell. They connect in the bundle sheath cell. And also this another mesosphere cell I attaching. And so currently, last Friday, I finished quantifying, counting how many cells, how many plasma desmata connecting each other cells. So hopefully we can see what's any difference in the cell. So, but here is problem. So in earlier I mentioned I have more than 200 trenching line. So working with the SBF-FM is not SBF is not one-side feed or microscope, because it takes such a long time to do the 3D image processing or some sampling preparation. So we are, based on this SBF SEM image information, we apply for the LSM 8080 with the area scan image. Because we know about the information of a chloroplast, we can optimize sampling preparation methods. So this is the previous our LSM image. One of the big issues is because if the chloroplast too much close each other, and the computer cannot distinguish individual chloroplasts, so as you can see, like this big the chunk of the mesosphere chloroplast. So after, based on this information, we are optimizing the microscope analysis, and then we can see, even you can see the two-dimensional image clearly, one the individual image, and as also you can see the clear image into LSM in the mesosphere cell chloroplast in the LSM image. And also this is application, so I'm trying to apply for this optimized method to the trenching line versus we just comparing. So one is wild type, and the other one is trenching line 1. This is trenching line 2. So this one, we can't see any difference, but trenching line has something like a different the chloroplast shape than the other one too. So, and also we are cross-check the data obtained from the LSM 8800 versus SVF SM data. They are almost the data is the chloroplast volume is the average. It's like they are overlap, so they are completely comparable. So we can use it to choose it. So that's my application for LSM 8080. And so this is summary. So using the SBF SEM, so chloroplast bundle sheath cell in sipo plant has a larger and more densely packed chloroplast in mesosphere cell, and both sugarcane and maize. And also we observe the plasmodesmata. They are linking the mesophyll and bundle-shell cell, and we're trying to quantify this information in C4 plant. So, and also regarding the method, so chloroplasts, in case of chloroplast estimation, the SBFF pretty consistent, less variation, but LSM is have a little bit variation because but LSM is much more faster. In case of LSM method, in our method, we can sampling preparation just like 5 minutes, and then images capturing only 10 or 50 minutes. So total 20 minutes, we can get the image and the overall information. But in case of SFM, you need to spend 7 days sample preparation and segmentation. I cannot say it's like take such a long time. So there are some like pros and cons. And so when you do your experiment, you can choose which microscope is good and works for your research. That's it. So that's all my research presentation. So thank you for your attention. And also this is our lab member picture. And also, as I suggest my advisors, I put my mesophyll cell in different color with the IGB. This is my final goal in this research. So thank you for your attention.
So are there questions?
Yep. Yeah.
That was fantastic. Thank you. Can you talk about the sample preparation before you do the microscopy?
Which?
How do you have to, I guess, the first microscope that you talked about?
The SVF-SM.
Yes.
So SVF-SM is, Kingsley is more expertise than me. So I just simply tell you like, we just have some fixing solution. We just cut the sample, the live leaf sample, cut the live leaves and put them in the fixation solution. And then we do some fixation and then drying, and then we just like using the resin stuff. So we just like fixing, and then we mount and look at. So.
Do you think that any of that and the fixation process has an impact on what you see?
Yeah, that's yeah. So I think it seems like the Professor Lisa answers asking about the sampling issues, sampling during one of the. Actually, I talked to Kingsley, but Kingsley is not agree with my idea. So, but SVF, SM, and TM, this stuff is they do the dehydration step. It seems like Professor Lisa answers worry about the shrinkage or some sampling distortion about this issue. That's she point out. So I didn't show the all of information this slide because we are currently we are trying to publish this data. So that's the reason we are concerning or same thing we are concerning about it. So we just collecting this data and we are LCM and just taking the live just like a regular microscope. We're just comparing all of them. There might be some shrinkage issue, the volume shrinkage issue, but still the value is still comparable with LSM the microscope. That's the reason we still reasonable shape, reasonable number, this thing. But Kingsley is not agree with any shrinkage issue, so I think he said because this is more traditional, I mean, more people using the TEM and all people using the same sample processing method. That's the reason. Yep. Yep, Sambon.
Yeah, thank you for the talk. I have two questions. One is curious about technical. Would you label the chloroplast and the salt the same as parts or something instead of using the microscope? Would you do that one or not? Or is there any technical difficulty to label the cells?
Someone, I couldn't hear your question. Sorry, so you asked him the labeling about the chloroplast or?
Would you, like, when you create the transgenic, would you label the chloroplast with some GFP or something and sort the cell with using the fax or something and stain the cell separately instead of following the machine? Maybe that's a kind of a little bit high scope you've done, just taking the image. Is there any difficulty in C4, like to do this cell sorting?
Yeah, it seems like your question is like, what about using not the GFP and other like fluorescent something? I didn't try it, so maybe I'm not sure, but you maybe talk to the Dr. Sibu in the Alma building because I had some experience with using the flow cytometry machine. You can maybe sorting by the flow cytometry and other machine to measuring the size of the cell maybe, but I didn't do any work in experiments. So I don't have any experience about the labeling system.
Another thing is about, I wonder about the interpretation of like having larger chloroplast in the blended sheet as compared to C3. In the method you said that, okay, the larger surface area is used to capture the light.
Oh, so okay.
Yeah, in that case, in C3, like, it's known that the C3 blended sheets could also sense like light directly, and also in C3 plants they could use like photoscopy for chloroplast movement to increase the light diffuse efficiency. So, do you know if there is some signal transduction difference in C4 plant for the interpretation, and also if the chloroplast still move to capture the light in C4 plant also? If so, why they don't use the chloroplast movement instead of increasing the size? Do you have some idea?
I think I just make sure if I correctly understand your question. So one of your question is, so inside of the chloroplast, if a bigger, your first question is like they are moving or beneficial. So yeah, also the other publication, the simple plant chloroplast also moving based on the different light intensity. But in this side, one of I want to say is like if this mesophyll cell is bigger, they block the light in the bundle sheath, in chloroplast in the bundle sheath cell. That's also this chloroplast in the bundle sheath also they need light. They require light. But in C3 plant, they don't have any. Not all, but they don't have a bundle sheath cell. But rice and wheat, they have a bundle sheath cell, but they do not do any photosynthesis. They don't need light, so they don't need to worry about some size. They're bigger and larger. And also, the size versus the chloroplast movement relationship, I need to think about it. But yeah, but I'm not about. I don't have any idea for movement. But something definitely, if it's too much large, disadvantage chloroplast movement during the light changing or something. Yeah. Yep, Mitch.
What's known about the cell wall composition between the bundle sheet and mesophyll cells? Is there big differences?
I'm not.
Could account for the change in shape.
Yeah, but yeah, I need to look at. But yeah, I'm not sure about the composition about the bundle sheath or mesophyll cell. Yeah, because I'm working with the C progress as a bioenergy stuff. But yeah, I didn't think about the cell structure with the composition. Might be some difference. Yeah. Yep. Yeah, I don't have an idea, but Ryan, yeah. Yeah, I don't have any answer idea. So yeah, I just talk to later about this thing. So yeah, I can happy to answer your questions. Yep.
So one way to look at that would be to compare your Z-stack images in the 880, the Ellison 880, and the serial block fix, right?
Yeah.
And if you see, because those changes in shape were large.
Uh-huh.
So you would see those. So if you did the two types of images, you should be able to compare them. If like the fixation was causing a problem or not.
Okay, I will look at the slide image. Yep.
So did you quantify when you did the analysis of
Yeah, that's the good question. Don also asked me about when I presenting this slide every time. He asked me, where's the vacuum and where's the other space? But in this case, I can see only the chloroplasts and cell wall I can quantify. So in this, I didn't. First thing, I'm sorry for the presenting this kind of the partial data because I'm not putting in all of the quantify the information. So I will let you know the later individually. So we just basically currently what we got the information. I got the information of a cell volume and plasma desmata information, and also regarding the chloroplasts and the cell volume, we can quantify the volume by volume, how much occupy in the inside of the cell. That we have it, the information, and and also the surface area versus volume information. We got it, but we don't have any particular specific other component information. Yeah.
I thought you had a high resolution serial block face image somewhere.
Yeah.
Oh, yeah.
Yeah.
Can you find the other cell? If you go through a stack like this, can you find the others? Parts to the
Oh yeah. So I mean, not all. I mean, some slides catch some vacuole or some nucleus or some other component in anatomy and stuff, but sometimes it's very hard to tracing or manually do the segmentation work. So in this case, I'm just focusing on the chloroplast and cell wall because I think Kingsley only know it, how much he spend time to do the segmentation work. So that's I don't want to spend like an entire life in the computer. So this time I'm just trying to do focusing on the cell and cell wall chloroplast, trying to publish it. That's my first goal. So yeah. Yeah, there are a lot of information in this image. So I'm very fast this day. I'm just thinking about working with other persons, specifically this day. The AI system is very interesting, specifically Dr. Andrew Leakey's lab. They do some machine learning program. They counting the stomata density or stomata size, shape. So it might be good for tracing. The machine can counting the chloroplast or cell wall. They can distinguish, they tracing, they can detecting. Much more save my time because this image, I got the 500 slide. In my case, this day I'm using HI, I call human intelligence, so me. So it's very painful. So not efficient than AI. So that's my future goal, this system. But this system, main purpose is trying to understanding of a seafood chloroplast and cell wall and trying to, based on this information, and we got some plasma desmata information. So next step is trying to invest more time and energy in order to this data file. And also we can targeting the plasma desmata feed hole or not, modifying the gene for the future research. That's our goal in Don's lab. That's I can say it in this presentation. So yeah. Yep.
So this is a question I asked you before too. Do you yet have an estimate of how many chloroplasts there are in an average bundle sheet cell versus average?
This is another some problem. Like first thing is one of the limitation for my experiment. One is the sampling depth. First time previous the literature when I read it, they say 25 micrometer sampling depth enough to capture entire cell in C3, all of C3 plant. But in case of a C4 plant, I cannot capture entire. That's another limitation. But within our sampling depth, within the volume per volume, I counting. So how many chloroplasts per the cell within our sampling depth volume? So we did, yeah. I and Ryan did. So you could, just for example, bundle sheet cell in case of sugarcane, they have average 25 chloroplasts per in our volume, and mesophyll cell they have only 12 and 13 chloroplasts in the mesophyll cell. They are currently in their counting in this data file. Yep.
Just a comment. I came from a metrology institute.
Yeah.
Another country, and the problem about counting, cell counting, are being discussed. So I think it's amazing your work, but it is just a suggestion. When we were discussing how to measure, how to count, you just have to think about the size, it's about I'm counting numbers,
Mm-hmm.
And you were trying to count by different methods. I'm not a microscopy
Yeah.
researcher, but I remember that we have a microscopy method.
Mm-hmm.
And also with the facts, it's a good idea to try to cell counting by the facts. And also DNA. We were trying to compare between methods.
Mm-hmm.
And for example, for the chloroplasts, we can try to find, I'm a molecular biologist, I work with
Yeah. Mm-hmm.
stuffs, and we can figure it out as some kind of genome or plasmid that we can see that is one number,
Mm-hmm.
and then compare this. But for microscopy, they were trying to, if you try to do between different methodologies, you also see a challenge for you. It will be another discussion. So my question is, how many samples do you take for a good statistical?
Oh, at least this in this case, because there's some limitation of the sampling, so we collect the 3 replication each species. We have collecting 3 three replication from maize and 3 replication sugarcane, and also we take 3 images from per sample, and then we just took one of the best, the capturing site. We just take out subsample and just do the analysis.
And maybe you have some difference between the age and variety.
Oh yeah, yeah, yep,
So
yep,
that's a big challenge.
yeah.
Yeah.
Let's think.
Thank you, thank you.
Okay, so we have a little bit of pizza left if you want to pick up a little bit on more. And feel free to come up and ask us questions about it. But that data is some of the best data from the cereal broadcast.
Everything is like hugely helped me. So